I am going to explain you regarding biomolecules topic. These are the molecules which are related to the biological organisms, and these are large in their size, heavy sized particles. We are not supposed to call them as particles also. These are the huge molecules which are related to the biological systems, and these biomolecules, all of them are polymers. In turn, these include carbohydrates, proteins, vitamins, hormones, nucleic acids and enzymes. Based on all these topics, we will get minimum of 2 questions in our competitive exams. Now, firstly I will explain regarding carbohydrates. These carbohydrates, name itself it is saying hydrates of carbon. So, we can expect the general formula CxH2O taken Y is the general molecular formula of carbohydrate, but all the time necessarily this may not be the molecular formula only for the carbohydrates. There are certain molecules which are having this molecular formula are also not the carbohydrates. Examples for the carbohydrates, first I will write here C6H2O taken 6 means C6H12O6, C12H2O taken 11 times, C12H22O11, it is glucose and it is sucrose. Molecules which are having this formula which are not the carbohydrates, now I will write here C2H2O taken twice means CH3COOH, it is acetic acid. Though it possesses the same molecular formula, but it is not the carbohydrate. We can say general molecular formula CxH2O taken Y is the molecular formula for carbohydrate, but carbohydrates are only possessing this molecular formula is not the correct thing. Now, based on the source, based on the hydrolysis ability, we will discuss regarding the classification at first. Regarding carbohydrates, we will discuss now. As I said, these are the hydrates of carbon. These consist more functional groups, polyfunctional groups. They are hydroxy and carbonyl. Both the functional groups are present in these carbonate. These can be called polyhydroxy carbonyl compounds. As I am saying the word poly, many hydroxy groups, 2 to 4, at least 2 to 4 hydroxy groups are present in these carbohydrates. These can be classified into various types based on hydrolysis. Based on hydrolysis means if carbohydrate is hydrolyzing based on the units that are produced, these can be classified into monosaccharides, oligosaccharides and polysaccharides. Monosaccharides is the word which consists mono and saccharide. In all these three, saccharide is the word that is present. In Latin word, saccharum means taste to sweet, nothing but sugar. Monosaccharide, these cannot be further hydrolyzed. These are again 2 types based on the carbonyl functional group, aldoses, ketoses. Aldoses contain main functional group aldehyde, ketoses contain main functional group ketone. In turn, based on the number of carbon atoms, these aldoses are aldotriose, aldotetrose, aldopentose and aldohexose. Here our main topic comes glucose, which comes under aldohexose. Glucose is nothing but aldohexose. It contains 6 carbon atoms and aldehyde as the principal functional group. Next, ketoses. Same like ketotrioses, ketotetrose, keto pentose, keto, hexose is like. Keto, hexose, here fructose, which contain ketone as the principal functional group, and it is also containing the 6 carbon atoms. Glucose, fructose. Regarding these two topics, we will discuss in detail in this topic. Next, oligosaccharides. Oligo means few. Oligosaccharide when hydrolyzed we will get 2 to 9 nearly monosaccharides. As I said, monosaccharide may not be further hydrolyzed, and we are getting 2 to many monosaccharides by the hydrolysis of oligosaccharides, because oligosaccharide itself, it is formed by the combination of 2 or 3 or 4 based on the units that are present in those oligosaccharides. Best example for that, types disaccharide, trisaccharide, disaccharide, sucrose, maltose, lactose are the 3 important disaccharides which we will face in this topic. Sucrose, maltose, lactose are the disaccharide which hydrolyzes to produce 2 monosaccharides. Trisaccharides, the word itself it is saying hydrolyzes to produce 3 monosaccharides. Raffinose is the example for trisaccharide. Next comes polysaccharide, in the name itself poly. When it is hydrolyzed, it is producing monosaccharides 9 to many or 10 above. Examples for these polysaccharides are starch and cellulose. Starch and cellulose including glycogen. Glycogen also comes under polysaccharide. This is one of the simplest classification based on the hydrolysis. Monosaccharides, oligosaccharide, polysaccharides. Next, another one based on the physical properties. Physical properties based on their solubility, based on their taste, based on their physical appearance, based on the 3 categories. 2 types are present, sugars, non-sugars. As the name is saying, sugars, taste too sweet, crystalline, very much soluble in the water. Non-sugars, they are amorphous, means powdery like substances, sparingly soluble in water, and they won't taste too sweet also. Next, based on reducing ability. Here comes the important point, reducing ability. Polysaccharides, monosaccharides, oligosaccharides, whatever may be the saccharide, if it is having the ability to reduce Tollens reagent and Fehling solution. Tollens and Fehling. If they are having the capacity, if the carbohydrate is having the capacity to reduce Tollens and Fehling's reagents, then that is said to be the reducing sugar. Fortunately, all the monosaccharides are reducing. Whereas disaccharides also reducing except, it's a very important point, except sucrose. Sucrose is the disaccharide which is the non-reducing sugar. Polysaccharides, all of them are non-reducing. I would like to stress the point here once. Carbohydrates which have the capacity to reduce Tollens and Fehling's reagents, they are said to be reducing sugars. In our syllabus, what we are specifying those disaccharides, sucrose, maltose, lactose. Sucrose is the one which is the non-reducing sugar. Maltose, lactose are the reducing sugars. All the polysaccharides are non-reducing sugars. Then one more continuation of this point, glucose having the ability to form silver mirror with the Tollens reagent, and it forms red PPT with the Fehling solution. After the reaction with the Tollens reagent, silver mirror is formed with the glucose and red precipitate is formed with the Fehling solution. It is another type of classification, more stressable aspect based on the reducing ability. Another aspect in this, the existence of optical isomers in these carbohydrates. Optical isomers. Formula 2 to the power n. N is nothing but the number of asymmetric carbons or the chiral carbons One should know asymmetric carbon is the carbon which is having all the elements of symmetry to be absent. Elements of symmetry is absent. Up to your intermediate level, if you remember, carbon valency is satisfied with the 4 different groups, we can consider it as asymmetric carbon. I am specifying the word. Asymmetric carbon is the carbon which is having 4 bonds with the 4 different groups. If 4 different groups are present around the carbon, then we can call it as asymmetric carbon, or another word chiral carbon also. So, this kind of chiral carbons is nothing but the N. As the number of chiral carbons are 2, we will get optical isomers 2 to the power 2. 4 optical isomers we will get. In our glucose, chiral carbons are 4. So, optical isomers are 16. Number of optical isomers in glucose are 16 based on this formula, and how we will get is not in our syllabus. Based on the existence of asymmetric numbered carbons, we will get the optical isomers. Glucose, it is having 2 to the power 4, means 4 asymmetric carbons are there here. I will show the asymmetric carbons how these are present, and then number of optical isomers in glucose are 16. We will discuss regarding the structure of glucose now. Glucose is the aldohexose. As I said previously, it is having aldehyde as the principal functional group, and 5 hydroxy groups are also present. Based on this information, we can write like this: CHO, CHOH taken 4 times CH2OH. It is the molecular formula C6H12O6 can be represented like this. Existence of carbonyl group and polyhydroxy groups, in turn aldehyde group and 5 hydroxy groups. Structural elucidation with some chemical reactions. With hydrogen iodide, whenever glucose is on reaction with the hydrogen iodide, we are getting normal hexane. It indicates all the 6 carbon atoms in the glucose are in a straight chain. N-hexane. It is a glucose structural elucidation on reaction with hydrogen iodide, N-hexane is formed. It indicates all the 6 carbon atoms are in a straight chain. On reaction with hydroxylamine and hydrogen cyanide, we are getting oxime and cyanohydrin. Oxime and cyanohydrin, these two indicates the existence of carbonyl group in the glucose. And on reaction with, as I said earlier, with the tollens and phenylanes, as it is having the capacity to react, we are confirming the existence of aldehyde group. And it can be shared on reaction with bromine water, we are getting gluconic acid. Gluconic acid, unless the presence of aldehyde, gluconic acid may not be the form. Glucose, when it is on reaction with nitric acid, we are getting saccharic acid. Glucose, when on reaction with these, what we are getting the products, it is the structural elucidation for what I said here. Existence of one carbonyl polyhydroxy groups can be sure on reaction with hydrogen iodide, getting normal hexane, means all the 6 carbon atoms are in a straight chain. On reaction with hydroxylamine and hydrogen cyanide, we are getting oxime and cyanohydrins. On tollens and phenylanes, it is giving a reaction, means it is responding with these reducing agents, we can be confirming with the existence of aldehyde group. When glucose is on a reaction with the bromine water, we are getting gluconic acid, it indicating the presence of carbonyl group as aldehyde, and on reaction with the nitric acid, saccharic acid is formed. It is indicating the existence of one primary alcoholic group, this one. This can be proved with the reaction with the bromine, and it can be proved, existence of primary alcohol can be proved on reaction with the nitric acid. It is the structural elucidation. By knowing this, we can write the open chain structure for the glucose. Open chain structure in the form of Fischer projection formula. It is the vertical Fischer projection formula. As I am symbolizing it in the vertical form, we can call it as vertical open chain vertical Fischer projection formula for the glucose. Here the numbering for these glucose 1, 2, 3, 4, 5 and 6. As I said regarding the number of these optical isomers, as I said the existence of 4 asymmetric carbons, here comes asymmetric carbons, second carbon, third carbon, fourth carbon, fifth carbon. C2 to C5 are the asymmetric carbons. As the number of asymmetric carbons are 4 here, we are having 16 optical isomers. Open chain structure of glucose is not satisfying all the properties. It is unable to react with sodium bisulfite and ammonia. It indicates the open chain structure is not justifying the structure of glucose. So, we will write closed structures, nothing but ring structures for the glucose. Now, I am writing ring structures for the glucose. It is the open chain. It is simple to remember, C2 with OH right side, C3 with OH left side, C4 and C5 OH groups are on right side. It is the open chain structure. Now, ring structures for this glucose. Ring starts from first carbon to fifth carbon, mediating with one oxygen. Means it is a 6-membered ring which contains 5 carbon atoms and 1 oxygen atom, and it is one of the ring form of the glucose. Another ring form same as like this from C to 3. Ring is formed in between C1 and C5. Apart from the first carbon, leftover configuration for these two forms will be the same. Now, I will give the name for these compounds, these structures. It is alpha glucose and it is beta glucose. These are the two ring structures for the glucose. Alpha glucose, beta glucose differ their configuration only at the first carbon. If you observe, on first carbon OH group is present on right side, there OH group on first carbon it is present left side. If OH group is present on the first carbon right side, we can call it as alpha form. If OH group is present on left side, we can call it as beta form. Apart from this alpha glucose, I would like to elaborate the name of this as alpha D plus glucose. So, I am writing these two terms extra. Of course, here also beta D plus glucose. Now, I will explain what is meant by this D and what are these plus forms. D and L, here the D what I wrote there, it indicates the absolute configuration. Absolute configuration includes D and L forms. These two are enantiomers. Enantiomers are the optical isomers which are non-superimposable mirror images. Primary condition to exhibit D and L configuration, the compound should possess at least one chiral carbon, and that chiral compound structure resembles with the glyceraldehyde. What I am writing here, the glyceraldehyde. Here it is the principal molecule to decide whether the given structure is D form or L form. If this kind of structure, if we can observe at asymmetric center, we can give the names D and L. Asymmetric carbon with OH group right side, we can call D form. Asymmetric carbon with OH left side, we can give it as L form. So, the same thing we can observe here, C1, C2, C3, C4, C5, C6. In these C2 to C3, C4, C5, 5 asymmetric carbons are here. Highest numbered asymmetric carbon with OH right side. Highest numbered carbon with OH right side, further here it is D form. Same as here, highest number OH on right side, further it is D form. If it is having the OH highest number with left side, then we can call it as L form. And the plus form is nothing but whenever asymmetric carbon containing compound is facing with the monochromatic light. If monochromatic light has fallen on the substance, if it is able to, the substance or the compound, if it is able to rotate the monochromatic light towards right, then we can call them as dextrorotatory. If the substance or the compound rotates monochromatic light towards left side, then we can call it as levorotatory. As these molecules are having many asymmetric carbons, these are also having the ability to rotate monochromatic light. Here the glucose is having the ability to rotate monochromatic light towards right side. For that we are giving plus. Dextro is nothing but plus, levo is nothing but minus. As it is dextrorotatory, we are calling it as plus glucose. Don't get confused with the capital D with the small d. Dextro can be represented small d or plus. Capital D is the absolute configuration, asymmetric carbon with OH right side. Small d which shows optical rotation when monochromatic light is formed. If it is having the ability to rotate towards right side, then we can call it as dextro. Here I think so now you got alpha D glucose means alpha form first carbon with OH right side, D asymmetric number, highest asymmetric number with OH right side, plus as it rotates plane polarized light towards right side. This is the information that you have to know. Here the questions may come like, what is the reference molecules to tell D and L forms, or the D and L enantiomers? Glyceraldehyde is the reference molecule. Here the asymmetric carbon with OH right side, D form, asymmetric carbon with OH left side, L form. And how can you say this dextro levo means ability to rotate the plane polarized light or monochromatic light? Scientist Haworth proposed pyranose structures for these alpha and beta forms. Alpha form of the glucose, as I said, C1 with OH right side, C2 with OH left side. This is the alpha form. Truly it can be represented. This can be it's an open chain. First what I wrote, now I am writing a ring form. It is a ring form of the glucose. Now it can be shown as Haworth structure. It is similar with the pyron. So the name of this compound is pyranose. Ring is formed, 6-membered ring. First carbon, second, third, fourth, fifth, followed by sixth. Now oxygen is present in between 1 and 5, 2, 3, 4, 5. The groups which are present right side we can write at the bottom, OH, hydrogen F, second with OH right, third with OH left, fourth with OH right, fifth, though it is present left side, we have to write inside the ring because CH2OH is present outside the ring. It is the pyranose form of the glucose. Same we can write for the beta form also, it is alpha form. If we change the position, like if OH group is present at left, our groups will be the same. If OH is at right side, alpha form. If the OH is at left side, it is the beta form. These are the Haworth structures or pyranose forms of the glucose. Existence for the cyclic structures or ring structures can be proved on reaction with methanol due to the formation of methyl alpha glucoside, methyl beta glucoside. And one more important, for the existence of two forms of the glucose can be known by knowing mutarotation. Mutarotation is nothing but whenever glucose is crystallized from hot concentrated solution at 30 degrees centigrade, we are getting alpha form whose melting point is 148 degrees centigrade and showing 111 specific rotation. Plus specific rotation means it is having the capacity to rotate monochromatic light towards right side. If the glucose is crystallized from saturated aqueous solution at 98 degrees centigrade, we are getting beta form whose melting point is 150 degrees centigrade and showing the specific rotation 19.2 degrees. Whenever the glucose is crystallized from saturated aqueous solution at 98 degrees centigrade, beta form is we are getting whose melting point 150 degrees centigrade and it is showing the specific rotation as 19.2. When these two are separately taken in aqueous solution at normal temperature, both of them are changing their rotation and showing the constant rotation. Both of them are showing 52.5 degrees of rotation plus. this change in specific rotation of two different forms at equilibriums to 52.5, it shows the existence of two different forms. At equilibrium, the mixture of alpha form is 36% and the beta percentage is 64%. One should remember, when it is crystallized from hot concentrated, it is showing 111 degrees of rotation. When it is crystallized from saturated aqueous solution, it is showing 19.2 degrees rotation. At equilibrium, when they are separately taken in aqueous solution, both of them are changing their specific rotations and constant rotation both of them are exhibiting, that is 52.5. At equilibrium, the percentage of alpha and beta are alpha 36%, beta 64%. When the monosaccharides is on reaction with dilute bases, dilute alkalis, they are isomerizing among themselves. D-glucose can be isomerized to D-mannose, it can be isomerized to D-fructose. Reversible isomerization is possible on reaction with the dilute NaOH. D-glucose, D-mannose, D-fructose, these three are formed when these three are the isomers. Reversible isomerization or reversible isomers are formed when any of these three compounds on reaction with the dilute NaOH or dilute alkalis. This reversible isomerization is called Labrie, de Bruijn, Van Einstein rearrangement. Because of this reason only, fructose is behaving as reducing sugar. It is one of the most important point. Fructose, though it contains ketone as the principal functional group, when it is on reaction with dilute base, it isomerizes either as mannose or as glucose and it shows reducing ability with tolerance and failing solution. Because of this isomerization only, fructose is behaving as reducing sugar. Now we will discuss regarding fructose. It is a fruit sugar as it contains ketone as the principal functional group. Molecular formula same as C6H12O6. We can write open chain structure, ring forms for these also. Open chain structure is CH2OH. On second carbon we are having ketonic group. Third, fourth, fifth, sixth will be the same as glucose. Third with OH left, fourth and fifth with OH right side. This is the open chain form. As I said, these are the asymmetric carbons. Highest number asymmetric carbon with OH right side, for that it is D. And as it contains 3 asymmetric carbons, it shows optical rotation also. Fructose shows optical rotation towards left side. Whenever monochromatic light falls on it, it shows its rotation. Optical rotation can be directed towards left side. So, it is D minus fructose. Here minus means it is showing levo rotation. For this, its name is also levulose. Why the levulose for the fructose? Means it shows its rotation towards, it shows its optical rotation towards left side. And it is the open chain structure, and we can write ring structures as I said for the glucose. Here we will write ring structures for this also. From second carbon to fifth carbon with one of the oxygen, means here the 5-membered ring, C3 with OH, C4 with OH right side. It is second carbon with OH right side. If I write second carbon with OH left side, then this is beta form. Left over will be the same configuration. Now it is alpha D minus fructose. Here it is beta D minus fructose. By knowing the ring structures for the glucose and fructose, now one more important aspect. We observed the configuration in glucose differing at first carbon in alpha and beta forms. C1 with OH right, we called it as alpha, with left we called it as beta. As the configuration at the first carbon they are differing, they are called anomers. Whereas in fructose they are differing their configuration at the second carbon. C2 with OH right side alpha again, C2 with OH left side beta, but the configuration they differed at second carbon, for that these are called epimers. As the configuration only differs at the first carbon, we can call them as anomers. If they are differing their configuration at the second carbon, we can call them as epimers. Now I will discuss regarding disaccharides. As I said at first, disaccharides hydrolyze to produce two monosaccharides. In our syllabus, sucrose, maltose and lactose are present. Sucrose extracts from the cane sugar. When it is hydrolyzing, we are getting two monosaccharides. All these are having the same molecular formula, C12H22O11. When these hydrolyzes produces two monosaccharides, sucrose hydrolyzed to produce glucose and fructose. Same formula, C6H12O6, it is glucose, it's fructose. Maltose hydrolyzed to produce two glucose units only. Both of them are glucose units. Lactose hydrolyzed to produce same molecular formula for the lactose also. We are getting glucose and galactose. I have to repeat here once. All these are having same molecular formula. Based on the hydrolysis products only, we can predict. Based on the hydrolysis products, we can identify the given disaccharide. By seeing the C12H22O11, one cannot say whether it is the sucrose or maltose or lactose. If the disaccharide hydrolyzed to produce glucose and fructose, we can confirm it as sucrose. As the disaccharide hydrolyzed to produce only glucose units, then we can confirm it as maltose. As it hydrolyzed to produce glucose and galactose, we can consider it as lactose. The question may be asked in different ways. Disaccharide hydrolyzed to produce two functional isomers, then we can call it as sucrose, because glucose and fructose, both of them are having different functional groups, though they are having the same molecular formula. The question may be like, disaccharide hydrolyzed to produce two monosaccharides which are not enantiomers, which are not diastereomers, then we can simply predict those two monosaccharides will be the same, that is glucose. As disaccharide hydrolyzed to produce two epimers, I am repeating the word here, epimers, then we can consider it as glucose and galactose. How we can say those two are epimers means the configuration at fourth carbon. Here it is with right side OH, here in this galactose C4 with OH at left side. Of course, up to this level the question may not goes. If the question can be twisted in this, you can able to attempt. Disaccharide hydrolyzed to produce two epimers, then the answer will be lactose, because glucose and galactose are the epimers. Now in detail we will discuss regarding the monomers of these disaccharides and the linkage in between the monosaccharides. As I specified, the word linkage in between the two monosaccharides is called glucosidic linkage or glycosidic linkage. Previously it was asked once, glucosidic linkage or glycosidic, one and same. Glycosidic linkage is the bond which is present in between the two monosaccharides. Sucrose, here the monomers as I said just now, glucose and the fructose. Alpha beta glycosidic linkage is present in between the monosaccharides. In maltose, two alpha glucose units are present. Alpha 1,4 glycosidic linkage is present in between the monosaccharides. Whereas in lactose, beta D glucose is present and beta D galactose are the monosaccharides. In between these, the linkage is beta 1,4 glycosidic linkage. The specific linkages you should remember. In between the monosaccharides of glucose, alpha beta glycosidic linkage. Here alpha 1,4 glycosidic linkage. Here beta 1,4 glycosidic linkage. Of course, in these three glucose units are present. You have to be particular with in sucrose and maltose alpha glucose units are present, but in lactose beta glucose is present. Beta glucose is nothing but C1 with OH group at left side. This is regarding the disaccharides. And regarding the sucrose, one should remember one important point. I said at first sucrose is only the disaccharide which is non-reducing. These two are reducing. Non-reducing ability is due to the carbonyl group is blocked during the formation of alpha and beta. And one more specific point here, sucrose when it is hydrolyzed, it is forming glucose and fructose. Glucose, its specific rotation plus 52.4 specific rotation, here it is minus 92 degrees of specific rotation, whose value is more than plus. I would like to repeat the statement once. Sucrose, actually it is dextrorotatory, as it is having the ability to rotate the monochromatic light towards right side. When it is hydrolyzing, two forms are formed, one is dextro, another one is levo, but levo is dominating. So it is changing its specific rotation. This is called inversion of sucrose. Very important point. Inversion of sucrose, thereby it is called as invert sugar. Among these disaccharides, the notable point will be the inversion of sucrose and monosaccharides which are present and the linkages in between those monosaccharides. Polysaccharides. In day-to-day life, we are in touch with many polysaccharides. What we are consuming, the food, rice, wheat consists these polysaccharides. What we are wearing, the clothes, cellulose, thread, they are also polysaccharides. In these polysaccharides, we have to discuss regarding the starch, cellulose, glycogen. Starch, as you know, the source for this starch, as I said just now, rice, wheat, potatoes, vegetables like. Molecular formula for these three, C6H10O5 taken N. And regarding cellulose, from wood source, most of the cell walls are with cellulose only. Glycogen, here it is called animal starch. Here glycogen is deposits in liver and muscle cells of the human bodies or in animal bodies. Truly we have to know more regarding starch and cellulose regarding the chemical structures. General formula, as I said, C6H10O5 taken N. It is on hydrolysis. We are getting C12H22O11 N by 2 moles. So it is on hydrolysis. We will get monomeric units. Whether it is starch or cellulose, C6H10O5 taken in hydrolysis, we are getting disaccharides, further hydrolyzed to get monosaccharides. The important aspects here, starch in turn it contains two parts, amylose and amylopectin. Amylose, amylopectin in turn having alpha D glucose units. Alpha D glucose units are linked through alpha 1,4 glycosidic linkages. Glucosidic linkages. In amylopectin also alpha 1,4 glucosidic linkages are present. Along with this, one more important point. Amylose is a linear form, amylopectin is a branched form. The branch is formed in between the two linear structures, C1 of one branch to the C6 of another branch, C1 to the C6. I will show. Actually, as I said, it is 1, 2, 3, 4, 5, 6. Here 1, 2, 3, 4. 1,4 linkages are present in amylose, whereas in amylopectin the branch starts 1 to 6 of another row, and in turn it continues like what I said. in amylose. This point should be specific. The branch is formed between C1 and C6. Glycosidic linkages are in between C1 and C4. So, amylose is a linear form, amylopectin is a branched form. In starch, it is present 15 to 20%, it is present 80 to 85%. Based on the existence, the molecular weights may be 10,000 to 50,000. Here it is having more than 50,000 based on the number of units availability. Next, cellulose. Hydrolyzed to produce glucose, but the monosaccharides in the cellulose are beta D-glucose units. In between the beta D-glucose units also one linkage beta 1,4-glycosidic linkage or glucosidic linkage. This is regarding cellulose. The notable points or the specific points in the polysaccharides are the linkages among the monosaccharides. In starch, alpha 1,4-glycosidic linkage, in cellulose beta 1,4-glycosidic linkage. In between the amylose and amylopectin, the difference is the arrangement of these monomers. Amylose is the linear form, amylopectin is the branched form. The branch is formed in between first carbon of one row to the sixth carbon of another row. That is also the important point. It is regarding the polysaccharides. Now we will discuss regarding some possible questions from what I covered, and these are the possible aspects that you can get for the coming exams also. A dextrorotatory sugar present in fruit is. Most of the students will get confused. Fruit sugar is the fructose. Of course, fruit sugar is the fructose. If we face the options glucose, fructose, cellulose, starch, most of the students used to think fructose will be the answer. But you have to be very particular about the question dextrorotatory. Fructose is the levorotatory. So, answer is glucose but not fructose, because glucose is also present in fruits. 20% of grapes contain the glucose. So, the answer is glucose but not fructose. Next question. Invert sugar is. Invert. Actually, sugar is levorotatory. As I said previously, when it is hydrolyzed, it is forming two monosaccharides, glucose and fructose. Glucose shows a dextrorotatory, fructose is the levorotatory, but fructose levorotatory value 92 dominates over dextrorotatory. Means during the hydrolysis, as it changes its specific rotation from dextro to levo, we can call it as invert sugar. Now the invert sugar is levorotatory. I am repeating, sugar is the dextrorotatory. During hydrolysis, it changes its specific rotation. So now it is levorotatory. Next, anomeric carbon in fructose is. While explaining regarding the glucose, as said, the one which differs their configuration at first carbon, they are said to be the anomers. But the configuration here in fructose differs at second carbon. What I mean to say, where the configuration differentiating starts. Here anomeric carbon in fructose is C2. It does not mean if the configuration differs at the second carbon. You try to catch at C2 positions, from C2 position only the configuration is getting differed. So, C2 will be the first carbon with different in their configuration. So, C2 is the anomeric carbon in fructose. If the same question for glucose, the answer is C1. Anomeric carbon in glucose is C1, anomeric carbon in fructose is C2. Because C2 with OH right side we can call it as alpha, with left side we can call it as beta. Next, chiral carbon in glucose and fructose are. Chiral carbon is the carbon which is satisfying its valency with different groups. In glucose, 4 chiral carbons are present. In fructose, 3 chiral carbons are present. In glucose, C2, C3, C4, C5. I am specifying C2, C3, C4, C5 carbons are the chiral carbons, whereas in fructose C3, C4, C5. C3, C4, C5, C6 configuration in glucose and fructose will be the same. The two functional groups in a typical carbohydrate. It was the previous triple equation. Two functional groups in a typical carbohydrate. Means typical carbohydrate we can consider it as monosaccharide or disaccharide, whatever may be the saccharide. Here we are having two types of functional groups, carbonyl functional group and hydroxy functional group. One more aspect, writing molecular formula for high membered disaccharide. One should be familiar with writing disaccharide molecular formula C12H22O11. If I ask you to write for the trisaccharide, one used to get confused. How we have to overcome that confusion, I will tell you now. How we can write the molecular formula for trisaccharide, tetrasaccharide, C6H12O6 into 2 minus 1H2O. Whenever we are getting multiplication with the 2, we are going to get the molecular formula for the disaccharide by the loss of 1 mole of water. C12H24O12 minus H2O. As I said here, minus H2O. We will get C12H22O11. Like for trisaccharide, C6H12O6 taken thrice minus 2 moles of water. C18H36O18 minus 2 moles of water. Here C18H36 minus 4 hydrogens, it comes 32O16. Whenever they are asking you to write the molecular formula for the high membered disaccharide, just you multiply C6H12O6 with that number followed by N minus 1 H2O. For tetrasaccharide, C6H12O6 into 4 minus 3 H2O means C24H48O24 minus 3 moles of water. We will get C24H, 3 to just 6, 48 minus 6, it comes 42O21. This is how we have to write. Once previously trisaccharide raffinose formula was asked in AIEEE exam. The formula is C18H32O6. It may not be the simple to remember. If you remember this formula, multiplying with the number followed by subtracting less by one number of water molecules. If they are asking you to write about pentasaccharide, C6H12O6 into 5 minus 4H2O. Like we can write the molecular formula for the disaccharides. Another biomolecules, vitamins. From your school standard onwards, you are using, hearing, learning regarding the vitamins. Here also on competitive sake and in your intermediate public exam, the most important, easiest scoring aspect, vitamins. The word vitamin was coined by the scientist Funk. He awarded the Nobel Prize also. As the name itself saying vitamin, they are very much necessary for the growth maintenance. It controls the regular activities. Mainly we can get these vitamins through food only. There are different types of vitamins which are very much necessary for our regular metabolic activities. These are specially necessary for children, pregnant women and growing child also. These vitamins basically classified into two types based on the solubility. Fat soluble and water soluble. Fat soluble, water soluble. B and C are the water soluble vitamins. Leftover A, D, E and K are the fat soluble. Student used to get confusion. For water soluble they used to say A, D, E, K. For fat soluble they used to say B and C. But very simple hint to remember here, citrus, that is lemon juice dissolves in water. Lemon juice in turn contains C vitamin. So you can remember like that also. B and C are the water soluble. A, D, E, K are fat soluble. Next, another aspect in these vitamins, you have to remember scientific names or the chemical names, source, deficiency diseases. Vitamin A, its chemical name is retinol. The source, as you know from your school standard, again also I am repeating here, milk, butter, kidney, egg yolk, means the yellow part of that egg. If you eat much, you will get the vitamin A. Deficiency disease, if you are going to lose the amount of vitamin A in your body, you may get night blindness, redness in the eye also one of the symptom for the vitamin A deficiency. Next, vitamin D, it is called 𝗂𝗽𝗱𝗁, scientific name or the chemical name is 𝗂𝗽𝗱𝗁. Sources, as you know very much, from sunlight it is synthesized in the body. Of course, through food also we can get butter, liver of the animals, fish oils are the sources. Deficiency diseases, rickets in children, osteomalacia in adults. Those two related to the bones. Bones may become brittle and they may get pains during the joints also. Next, vitamin E, its chemical name tocopherol, and green leafy vegetables, oils, wheat are the sources for these vitamin E. Deficiency disease, sterility. Its sterility is nothing but unable to produce the next generation. Genetical disorders may come under sterility, and muscular dystrophy. As I said, for vitamin D, here also the muscles may not work properly. Muscular dystrophy is also the deficiency disease of vitamin A. Next, vitamin K, it is anti-hemorrhagic. The sources are cabbage, leafy vegetables, cow milk. We may get this K vitamin. If we are unable to get much vitamin K, we may get across the deficiency disease of excessive bleeding. If any scratch or cut on the body, the blood comes out continuously, and coagulation ability may reduces. Next, vitamin C. Its scientific name, its chemical name is ascorbic acid. It was previously asked in one of the competitive exam. Ascorbic acid is the scientific name for the vitamin C. Mostly it will get from citrus fruits, green leafy vegetables. Deficiency disease is scurvy, immunity may break down, resistance towards any diseases may decrease, absence of these vitamin C. Apart from these, a person may suffer from deficiency of one more vitamin or two more vitamins. That disease is said to be avitaminosis, multiple vitamin disorders. Now I will discuss regarding vitamin B complex also. Up to now what I cover regarding the vitamins A, D, E, K and C. Vitamin B includes complex, B complex, B1, B2, B6, B9, B12. B1, B2, B6, B9, B12. One should remember their chemical names also. B1 is the thiamine. Source for the thiamine, grains, nuts, nothing but milk, green leafy vegetables. Deficiency disease of thiamine is beriberi and weak heartbeat. Heartbeat fluctuations may be due to the deficiency of this thiamine. Next, B2, it is riboflavin. Source yeast, milk, egg. Deficiency is skin diseases. There is a darkness in tongue, red color of the tongue or soring of the tongue is due to B2 deficiency, that is riboflavin deficiency. Next, B6, it is a pyridoxine. Same sources, yeast, milk, egg. Of course, yeast you may get through the bread buns also. Deficiency, chronic anemia means there is a loss of blood percentage in the body. The person may get through anemia. And convulsions and vomitings. Convulsions means shiverings, vomitings as you know well, regarding the deficiency of B6, that is pyridoxine deficiency. Next comes B9, it is folic acid. Folic acid more available in spinach leaf, in it is Telugu called palakura, and intestinal bacteria while taking the non-veg, and deficiency diseases, growth retardation and anemia. For that reason only pregnant women used to consume lot of folic acid tablets folic acid is B9. Next comes B12, it is cyanocobalamin, and sources are liver, egg, curd. Deficiency, the percentage of RBC get decrease in the blood, nothing but the pernicious anemia comes, and nervous system disorders may be observed due to the deficiency of B12. One should be familiar with the chemical names B1 thiamine, B2 riboflavin, B6 pyridoxine, B9 folic acid, B12 cyanocobalamin. In cyanocobalamin, the metal ion that is present is cobalt ion, that is also one of the important aspect. After observing these many sources, one should eat, of course, especially those who are preparing for the exams, one should be touching with the green leafy vegetables, eggs and milk, and avoid the deficiency diseases due to vitamins. Now we will discuss regarding some questions based on vitamins. Ergo-calciferol is name of vitamin D. Second question, which of the following is water soluble? As I said, water soluble vitamins are B and C. Here the answer is B1, whereas A, D, E, K are fat soluble vitamins. So the answer is B vitamin. Next the question, condition of vitamin deficiency is known as I said, particular vitamin deficiency, particular diseases. If the person suffers from multiple vitamin deficiency disorder, then that is said to be avitaminosis. The question answer, to make you confuse, vitaminosis is also there, but avitaminosis means deficiency of vitamins. Next the question, ascorbic acid resembles the structure of. Ascorbic acid is nothing but vitamin C. Its structure resembles, the options are here, vitamin A, glucose, cellulose, vitamin D. If the answer is glucose, ascorbic acid structure resembles with the glucose structure.